is Carmen McIver, and I'm a program coordinator in the Office of Student Life and Engagement. I'm also the chair of the Women's History Month Committee, and as a chair, I would like to welcome you to our very first Woman Chat Wednesday speaker series featuring Dr. Lydia Komaroff. Mi Vida, Woman, Chicana, and Scientist is the presentation. And I want to thank you for joining us and celebrating Women's History Month with us. We all know that Women's History Month is not, or women's history is not just confined to the month of March, but of course, 24/7, 365. And we celebrate women from all over, and we benefit greatly from the contributions to from the women in our lives and around us. So it's a great day to be a woman. And again, I just thank you all for taking the time to celebrate with us and listen to a very powerful and also awesome woman who has been contributed greatly, to our world. So with that being said, I'm going to turn it over to my good friend Morgan, who's going to introduce our speaker.
Thank you. Hello, everyone. My name is Morgan Wright. I am the student ambassador here at the Mountain View campus. Let's get started. So, Dr. Lydia Via Komaroff. She is a molecular biologist, a business executive, and a diversity advocate. She was one of the early cadre of scientists who first used the ability to join DNA from animals or people to DNA from bacteria to make useful proteins and to study biological processes and human diseases. She has served on advisory committees for the National Institutes of Health, the National Science Foundation, and the National Academies of Science, Engineering, and Medicine. She represented small businesses of the State of Department at the Asian Pacific Economic Conference and serves on several academic advisory boards. She is a fellow of AAAS and the Association for Women in Science. She has been honored by election to the Hispanic Engineer National Achievement Hall of Fame, a Lifetime Achievement Award by Hispanic Business Magazine, the 2013 Women of Distinction from the American Association of University Women, and is the 2016 recipient of the Elton Morrison Prize for the MIT program in Science and Technology. She was one of 6 women scientists whose work was featured in the PBS series Discovering Women. Her work was the subject of a one-hour segment entitled DNA Detective. She was one of 11 women scientists profiled on the website of the White House Office of Science and Technology during the Obama administration. Please join me in giving a warm welcome to Dr. Lydia Via Komaroff.
Thank you, Morgan. It's a great pleasure to be here today. And let's see, am I sharing my screen? How did that go?
Yes, now you are. Mm-hmm.
All right, thank you. Thank you again, Morgan. I'm sorry that I can't be there to see who you are and meet you in person, but it's a great honor to kick off your series to celebrate Women's History Month with you. Today, I'm going to describe my journey and some of the lessons I've learned along the way. I'll start with my family and education, then talk about my career. I'm going to show you lots of pictures of the people who've been important in my life so that you can see that people who don't look like you and may not have much understanding of your background in life can really be important mentors. Jot down questions in the chat as I go along about me, my family, or anything as I talk, because I'd like to be able to answer those in the Q&A session. And there will be time for questions, and if some of you can, I can stay until 3:30. Like most Americans, I come from a diverse genetic background, mostly Spanish and indigenous Mexican, but also Arabic, African, Asian, and Jewish, with a little Neanderthal sprinkled everywhere. Although my European genes constitute the majority of my genetic heritage, I identify as Chicana, a Mexican American. My paternal grandparents immigrated from Mexico in 1915 during the Mexican Revolution. My grandfather, Encarnacion, had no European blood. He was puro indio, and my grandmother, Maria de la Luz, was of Spanish descent. To support his family, my grandfather came to the U.S. in 1906 to work on the railroad. At that point, Mexicans seeking work could enter the U.S. fairly easily. He was also an enterprising guy. After the Mexican Revolution started, he kept track of the battles between the Federales and the revolutionaries. He'd collect all the rifles he could carry and resell them. He had to leave Mexico in 1915 because the Federales were after him. They wanted him for gun running, and his family was at risk. So, with his two small children, his parents, his grandparents, his widowed sister, with her two small children, and his younger brother, the family trekked over 1,100 miles, stopping along the way to work for food. They ended up in Clovis, New Mexico, and my grandfather built this house where my father grew up. It had no running water and a dirt floor, but it was the family home. My dad was one of 15 and a middle child. Only 10 survived to adulthood, and everybody worked in the fields as children. All of my father's brothers played music by ear, and my dad played the violin, and he would earn extra money by playing on the street. A violin teacher heard him one day and taught him how to read music and how to properly hold the violin. My Uncle Max told me that Dad spent hours listening to classical violinists playing Beethoven and trying to get the same sound. My Uncle Max also told me it drove everybody else in the family crazy. My dad was a tail gunner on a B-17 in World War II, and he was able to finish college because the GI Bill paid his way when he came back from Europe. My mother's forebears on both sides of her family came to the New World from the Basque region of Spain with the conquistadores. My maternal great-grandfather, Manuel Larrañaga, could speak English, Spanish, and Apache, and he had 2 books, the Bible and a translation of Jewish history, that he got in 1913 and that my grandmother gave to me when I graduated from college. As you can see, he had beautiful handwriting. His wife, Cleophas, was a strong-minded woman. As a young woman, she once traveled 3 days by covered wagon to go to a party. Unlike most women back then, she didn't know how to sew when she got married, but she taught herself how when her husband gave her some beautiful fabric as a wedding present. She made my mother's christening dress, and as my mom grew up, she would make her clothes from pictures in magazines so that my mother was always in style. My great-grandparents had 10 children. Their sons joined their father on the ranch in Arizona as soon as they had learned how to read and gotten some basic arithmetic under their belt. But he sent his daughters to normal school after high school for teacher training because he didn't want them to marry cowboys. My grandmother was a single mom. She married another descendant of the Spanish conquistadores, but divorced him when he became an alcoholic. Like my mother, my grandmother worked most of her life. She moved in when I was in grade school and was really an important person to us. Both of my parents were college graduates. They were each the first in their family to go to college. I was born while they were in college, and one would take classes in the morning and the other one in the afternoon so that one of them could be home with me. Both of my parents grew up speaking Spanish, but because they had been punished for speaking Spanish in the school yard, never mind the classroom, they decided that their children would grow up speaking only English. Spanish, therefore, was the language of secrets for me, although I learned to understand it quite a bit from my great-grandmother Cleophas, my great-aunt Linda, and my grandmother. Today, many Latinx students grow up with English as a first language and may feel like outsiders in both cultures. I grew up in Santa Fe, New Mexico, the eldest of 6. My mother was a social worker. My father was a music teacher. My brother, Richard, is a Vietnam vet and was a policeman, an FBI agent, and a federal investigator for the Federal Public Defender's Office before he retired. Catherine is a judge in Oregon and was a public prosecutor before she joined a law firm. Roland was a musician and a music teacher. We lost him to pancreatic cancer when he was 48. Lawrence is a bilingual science teacher, now working with children with reading disabilities. And Dorothea was a bank executive and then director of compliance for Honda America. She got married right after high school. She eloped, in fact, and she and her husband went to community college and then transferred to a 4-year college to complete their degrees, working full-time the whole time. Her husband, Michael, ended up as a senior executive at Southern California Edison, and his expertise in electrical grid modernization was recognized worldwide. So when my mom and dad decided to go to college, and when their parents supported that decision, they changed many lives. We are a very large extended family. I have over 100 first cousins, 17 nieces and nephews, and 30 grandnieces and nephews. I decided I wanted to be a scientist early, about 9 years old, and by high school, I was the only girl in the physics class. I applied to the University of Washington because my father had a brother in Seattle. I started as a chemistry major, but had trouble with classes, including almost failing organic chemistry. When I went to see my advisor, he said, "Of course you're having trouble. Women don't belong in chemistry." I didn't question him. I just changed my major, several times in fact. And then I finally settled on biology, where several young and enthusiastic faculty encouraged me and expected me to do well. I retook organic chemistry and I aced it. As a sophomore, I met Tony Komaroff, a medical student who wasn't at all surprised that I wanted to be a scientist. So when he went to NIH after he graduated, I transferred to Goucher College. Not much is going to happen across the country if one of us didn't move. One of my professors suggested that I go to Johns Hopkins, but they didn't accept women back then, and Goucher was their sister college. There, I worked in Dr. Gardner Moment's lab, and he always said, "Cherish your exceptions," an invaluable lesson for a scientist. I got a summer job at NIH with Dr. Loretta Levy, one of the first women to run a laboratory at the National Institutes of Health. She was responsible for getting me into MIT. I had applied to every school in Boston except MIT because Tony had an internship there after NIH. When I asked Loretta for references, she said, "Where's MIT?" And I said, "I can't go to MIT. I'm not good at math." And she said, "You told me you wanted to be a molecular biologist. MIT has the best program in molecular biology in the nation and probably in the world. If you're serious about becoming a molecular biologist, you have to at least apply to MIT." Well, it's a good thing that I did because MIT was the only school in Boston that accepted me. It taught me you can't get what you don't apply for. Tony and I were married the day after I graduated and we moved to Boston. My class at MIT was small. There were only 15 of us. And in those days, we all took classes together and then we chose a thesis lab in January. Our class met as a group and we worked out between us what labs we wanted to approach for our thesis so that no two of us would approach the same lab. I went to go see David Baltimore, who worked on viruses and who later won the Nobel Prize for discovering an enzyme that copied RNA into DNA. He told me he didn't have any room in his lab. At the time, he and Harvey Lodish shared an open lab and office space, so I went to Harvey and proposed that I do a joint project with him and with David on the translation of poliovirus RNA and that my space be in Harvey's space. Well, at the time, poliovirus was being studied in David's lab and Harvey was interested in cell-free protein synthesis, so Harvey was very enthusiastic. So I went back to David, and that's how I became the first graduate student in biology at MIT to have 2 thesis advisors. Poliovirus is a small virus. It has 4 proteins in a coat shaped like an icosahedron surrounding a single RNA molecule that in an infected cell directs the synthesis of the 4 coat proteins and about 10 other proteins the virus needs. The virus RNA is about 7,500 bases long. For comparison, the insulin RNA is about 550 bases long and, like most mRNAs, codes for 1 protein. Let me just remind you that there are 4 chemicals called bases that make up an RNA. These are the letters of the alphabet for making proteins from the 20 amino acids, which are the building blocks of all proteins. 3 bases make up a word or codon that specifies one of the amino acids. Some amino acids are specified by more than 1 codon. We knew that in cells of all kinds, ribosomes, the protein-making machinery, start at the methionine start site, and slide down the messenger RNA, putting in the correct amino acid at each codon, stop at the stop sign, and release the completed protein. I should note that when poliovirus infects a cell, it stops the production of cellular proteins. Only viral proteins are made. So the question was, how are the separate poliovirus proteins generated from this very long RNA? There's three possibilities. Maybe the long RNA was broken up into smaller messenger RNAs, each coding for one protein. There might be, on the other hand, internal start signs that the ribosomes could recognize. Or maybe there's only one start sign and a very long protein is synthesized and then cut into the later proteins later. Remember, poliovirus infected cells are only making poliovirus proteins. And when we looked in poliovirus infected cells, we only saw the 7,500 base viral messenger RNA covered with ribosomes, eliminating the first possibility. So the question then was there a internal site at which protein synthesis could be initiated, or was one long polyprotein processed after translation? So if we go back and look at our model RNA, while methionine is always the starter amino acid of a protein, it's also found inside proteins, and it only has one codon. Fortunately, methionine can be chemically modified so that ribosomes can only use it at a start methionine and not at an internal methionine codon. So for my thesis, I used methionine that can be inserted into any methionine codon, and I see several different size peptides. But when I used the specially labeled, the modified methionine, I only saw one, indicating that there was only one start site. So with that, towards the end of my graduate studies, I was told by Harvey that if I went to this very big meeting, the FASEB meeting, I could later in the year go to the ski meeting. Well, that was a no-brainer, so of course I went to present my thesis work. There, for the first time, I met several other Mexican American and Native American scientists. I didn't know they existed, and we established SACNAS. The only other woman at that meeting was a member of one of the agencies. SACNAS is now 47 years old, and it has become the most diverse scientific society dedicated to attracting and encouraging young people from all groups historically underrepresented in science. When I graduated, I was glad that my parents, my grandmother, and great aunt and great uncle were able to attend graduation. And after I received my PhD, I moved to Harvard for postdoctoral work. In my field of biology, if you want to set up your own lab, you have to do an internship sort of after you get your PhD. So first I worked in Fotis Kafatos' laboratory on the development of the silk moth eggshell, where Argis Stradiatis was my lab mate. There were a lot of Greeks in that lab. My project depended on connecting moth eggshell DNA to bacterial DNA to isolate and characterize the moth eggshell proteins and genes. Now, this kind of work got banned in Cambridge because at the time it was believed that, these recombinants might be dangerous. People talked about Frankengenes. So I joined Tom Maniatis, who had moved from Harvard to Cold Spring Harbor Laboratories in New York because all of his work depended on this new technology. And for me, nothing worked for a year. This is really bad news because getting a job in a university and setting up my own lab depended on my having publications from my postdoctoral work. Meantime, Art was working with Wally Gilbert, who would later get the Nobel Prize. And as I was getting ready to return to Boston, Art urged me to join him and Wally in their effort to make an insulin bacterial DNA recombinant. Well, I hadn't gotten any publishable results at Cold Spring Harbor, but I had become very good at connecting DNAs, and I had been given a small sample of all the enzymes in the Cold Spring Harbor refrigerator, a priceless treasure. At the time, many of the important enzymes we used were not available commercially. We had to make them, and there certainly weren't any kits that we could use. So I joined the Gilbert team, and 6 months later, this has never happened again to me in my scientific career, we had not only made and characterized an insulin bacterial DNA recombinant, that recombinant was making insulin in the bacteria. This was pretty exciting, and I always try to remember that Wally would tell us it's just as hard to solve a trivial problem as an important one. So it's worth it to go for the important problem. The paper got a lot of attention, both scientifically and by the national press. Everyone knows someone with diabetes, and indeed, almost all insulin today is produced by these methods. The cost of insulin today is not because of the methods used to make it, by the way. So in order to write this paper and to do the experiments, we had to use a combination of pushing the limits on the technologies available to us, and we had to look for clever ways to modify old techniques that had fallen out of use. The very first thing we had to do was to identify a source of insulin messenger RNA. We wanted to use rats as a model because you couldn't use humans. The cells that make insulin are tiny islands of cells that are sprinkled throughout the pancreas, which is under the liver. A 1 pound rat, that's a big grown-up rat, will have a pancreas that's about 1/10 of an ounce in weight. So getting enough cells to make enough RNA requires many, many rats and lots of careful dissection. Not an optimal choice for impatient molecular biologists. So we turned to a colleague, Bill Chick, who studied diabetes in rats at the Diabetes Hospital in Boston. He had lethally irradiated rats, and then he saved those rats by connecting the circulatory system of each irradiated rat to the circulatory system of a healthy rat. This is an old technique. In one such pair of rats, a pancreatic tumor arose. That tumor could be propagated in other rats. So those tumor cells became our source of RNA. We knew that insulin RNA was only a few % of all the RNA in the cell, so we were going to be looking for a needle in a haystack. The next steps were all done in little tubes, like the one I'm holding here. We used a plasmid called PBR322, named for its builders at Stanford University, Francisco Bolivar, who was from Mexico, and Ray Rodriguez, a Mexican-American whose parents were farm workers. I broke open the plasmid at a specifically chosen site and added linkers to each end. Arj prepared RNA, copied it into double-stranded DNA in several steps, and added linkers to that end. We then mixed the plasmid and the cDNA. The complementary ends found each other, and we would get circles, some of which had an insert and some of which did not. DNA and RNA are really very flexible. They're more like ropes than like rods. We used PBR322 because it coded for 2 drug resistance genes, so that a bacteria infected with an intact plasmid with an insert would become resistant to one drug, drug 2 in this case, and not the other drug, drug 1 in this case. So then when we had mixed our bacteria with the mixture of plasmids that had been connected to inserts, we mix them under conditions where the bacteria swallow the plasmids, and then we spread out the bacteria on plates such that the bacteria lands spaced out from each other. And we grow them on the 2 drugs. We got 1,000, the colonies look kind of like this. And when we plated them, we got 1,745 clones on plates that look like this. Then we used a series of molecular biological techniques to identify the clones that had insulin, and there were 48. And that actually took most of the time of those 6 months I told you about. So once we had the 48 clones, I plated them on 2 plates, picking them off the plate, and then just stabbing the toothpick in a pattern on the 2 different plates. And it started snowing. It snowed because that was the blizzard of 1978, and all the roads were closed and nobody could go anywhere for several days. So my clone sat in the incubator. When it stopped snowing, I hitched a ride with my husband, the doctor, and then I hitchhiked over to Cambridge to the laboratory I was working in. And to my surprise, I saw that there was 1 clone that was growing on both antibiotics. And I knew that all the clones I plated had an insert because we had taken them and characterized them. So I immediately ran to the office of a friend, an eminent cancer biologist, and said, I have wonderful news. He looked up at me and exclaimed, you're pregnant. Ah. So then I went to go tell David Baltimore, and he got it right away. I immediately concluded that in that clone, we were making a hybrid protein where the protein that specified drug resistance was connected pro-insulin, and that turned out to be the case. So besides the Proceedings of the National Academy paper, Wally and I wrote a paper for Scientific American, and we got 3 patents. Those patents, by the way, did 2 things. They were the basis for starting a company you might have heard of called Biogen, and royalties from the patents provided some nice income in later years. Well, with that paper under my belt, I became an assistant professor at UMass Medical School in Worcester. Tony, meantime, was a faculty member of the Harvard Medical School, so we had solved the two-body problem that many professional couples face. 8 years later, I received tenure, despite having made essentially all the mistakes that women and individuals from historically marginalized groups. I was on too many university committees, I developed and taught more courses than most of my colleagues, and I didn't publish enough. I didn't know that I could say no, and so I neglected my primary responsibility as a scientist and a new faculty member, write papers. So a year after I was tenured, I accepted a non-tenured position at Harvard Medical School and Children's Hospital, and I had a wonderfully productive decade there with many collaborators and students and postdocs. And to my delight, a lot of these papers continue to be cited in the current literature. Now, I was better at balancing things at Children's and Harvard, but I still didn't say no often enough. And after 8 years, I began to feel I needed to change something. I thought I'd go back to the lab full time because I really loved that work. But I talked to lots of people, always a good thing to do when you're thinking about a major change. And I began to think that maybe it was time to move to a position where I could use my administrative skills and broad interests. As a result of those discussions, I got a call from a headhunter looking to fill a position as Associate Vice President of Research Administration at Northwestern University in Evanston and Chicago. My first reaction was, why would I go to Chicago? Tony's not going to leave Harvard, and I had only been to O'Hare. But the headhunter was another wise person, and she said, look, you don't have to take this job, but if you want a job like it, you have to look at this job. So I went to look. I met Bill Kern, the vice president and a theoretical chemist, and I met the administration and faculty members. So in the end, I took the job, and Tony and I started a 7-year commuter marriage. Bill Kern was a spectacular mentor in the administrative arena. 2 years after I became associate vice president, I was appointed vice president for research after a national search. I loved that job. It taught me to appreciate work in all areas of scholarship, and importantly, I had the resources to make strategic investments in faculty and students, thanks to President Beenan, who was a spectacular fundraiser. Don Jacobs, who was the Dean Emeritus of the Kellogg Business School, invited me to attend the 1-month 24/7 Advanced Executive Program, sometimes called MBA in a Box. That course was very important in filling holes in my business knowledge. After 7 years, it was time to end commuting and return to Boston. I had tried to recruit Susan Lindquist, a superstar, to Northwestern, but she ended up recruiting me to the Whitehead Institute, where she had become the first woman director. During my time as vice president at Whitehead, I joined my first corporate board, and a year later became chairman of the board, a story for another time. Susan decided to return to the bench after 3 years, and I moved to the business world in 2005, joining a company that had been started by Wally Gilbert's son, John, on the basis of a rapid microfluidic switch he invented. With it, he envisioned a microchannel chip that could be used to sort cells sterilely for cell therapy, and he'd started Cytonome to build such a sorter in 2002. Well, after 6 months, John and I switched roles. I became CEO and he became Chief Technical Officer, and then he decided to continue as a serial entrepreneur and go found more companies. Cytonome was the 3rd company he had started. So he did this in 2000. He made this decision about 2008. and this is where persistence really paid off. I had to pitch the company over 200 times during the Great Recession of 2008. And in fact, after one unsuccessful early round, I had to lay off half of the company. That was 30 people I had to fire in one day. And furthermore, the three of us executives who survived took no salary, and two angel investors and I, all members of the board, put in enough money to keep the company afloat. I used home equity. In March 2009, fortunately, we signed an agreement with a company based in Texas and formed Cytonome ST LLC as a subsidiary of ST. John Sharp, an electrical engineer from New Zealand with extensive experience in building cell sorters, became CEO, and I remained CSO and board member. ST's business model is the sale of bull sperm carrying the X chromosome. Juan Moreno and Maurice Rosenstein started ST to provide sexed sperm to dairy farmers. A dairy cow has to be replaced every 3 years. And so dairy farmers really don't want males, they want females. At the time, most breeders sold unsorted bull sperm to them. So Juan figured out how to sort bull sperm, and then he bought every used MoFlo, a particular kind of cell sorter he could find, and he put them all in a large facility in Navasota, which is just up the road from Houston. John, who had helped design and build the MoFlo, was a consultant to ST, and they were always on the lookout for promising technology, which is why we met them. So John immediately put the team to work building an easy-to-use, reliable sorter for ST, while continuing to develop the Gigasort, the therapeutic sorter. In May 2011, the first perm sorter was installed at ST, and we had a working Gigasort prototype by 2012 and could publish a description showing that it worked. In 2014, after 9 years, I had originally planned to stay at the company a year and 3 logos. I left management at Cytonome. In addition to Gigasort, by the way, the company's developed a prototype tabletop droplet sorter, an industrial cell sorter that can be used for specific uses, and has shipped over 300 sperm sorters all over the world for ST Genetics, formerly Sexing Technologies, and the Gigasort, the therapeutic sorter, is in use in Japan for a clinical trial using differentiated cells for the treatment of Parkinson's disease. So after a year of semi-retirement, but still being busy, I was tired of introducing myself as a former this, that, and the other. So I established a one-person company to be an umbrella for my activities. And I serve on boards and committees and give talks like this one. At this point, I'm focused on issues that account for the slow change in representation in colleges, universities, and companies, despite the rapidly changing demographics of the country, especially for me in the STEM fields. What I'm doing is introducing scientists, faculty members, administrators, company management, who tend not to know or appreciate the social sciences. I want to introduce them to the biological basis of decision making and how that can result in decisions that they make that perpetuate the lack of diversity, even though they may truly want to increase representation. These books were my first introduction to to human behavior, and Daniel Kahneman's in particular is important. He is a Nobel laureate in behavioral economics. So I'm going to give you a brief overview of that work. This slide summarizes the synthesis of work done by social scientists, behavioral neuroscientists, psychologists, and others. The human brain is quite remarkable, this little piece of organ up here. It has many cells. It's very complex, but it can't deal with all of the data it receives. This is the ultimate big data problem. We're constantly receiving input from all of our senses. And in order to be able to function in the world, the brain has to block or ignore much of that input. This is true of all brains, by the way. As a result, we've developed shortcuts that allow us to make decisions very rapidly. Gut decisions, we call them often. Some of these shortcuts are hardwired through evolutionary experience. If you're in the jungle or on the veldt, if something new comes your way or something's waving in the breeze and you haven't seen it before, it's very likely to be dangerous. So you react accordingly. And you may have seen videos of cats reacting to cucumbers. This is an atavistic reaction that may reflect an evolutionary response to the danger of snakes. Other shortcuts are formed in response to the culture that we grow up in. And all members of our culture are subject to these unconscious or implicit shortcuts or biases. And in order to change our behavior, we first have to acknowledge that the biases exist, and then we have to try to understand why they exist, which is to say, we need to know our history, the good and the bad of it. So let's talk about how they arise. All of you are familiar with optical illusion. Here's a very old one. You probably all see this as two tables of a different size. I see them as different sizes, and I know they are the same size. And I can show you that that's so by outlining one and then using the magic of PowerPoint, just rotating the rectangle and sliding it over to the other table. You can do this yourself. You can find the image on the web if you look up Shepherd, an image, and you can show that I didn't do any tricks. It really is the same size and it looks different instantly. This happens because we have a two-dimensional retina, but the world is three-dimensional. And so we've learned how we should interpret what we see. And as a result, some of the time we see optical illusions. Well, implicit bias stems from associative errors. We associate two things with something that they're not really associated with. And we learn these associations in two ways. Some we can learn explicitly by practice. For example, in order to play an instrument, musicians have to learn to associate what is essentially a pattern of dots on page to particular sounds and to where you will put your fingers on an instrument to make that sound. With practice, this becomes automatic. And when a musician, especially a good one, is playing or singing, when Beyonce is singing and dancing, she's not thinking about those notes. She's making music. And it's become the muscle memory we talk about is what she is using. Now, some associations are learned as we are exposed to our society. That means the movies, TV, our friends, our parents' attitudes, the people we see in different roles, scientists, engineers. Who do we see as engineers, judges, lawyers, police, etc.? Some of the associations that we make unconsciously may be counter to what we consciously believe about ourselves. Hence the constant refrain, I am not racist, when someone has just done something which is in fact pretty racist. This is because they believe and honestly to not be, they're not racist. They just have these habits of thinking because we all have them. And the most important thing I want to stress is that all humans of a culture share the same biases. So women are as likely to be judgmental in these biased ways against other women as are men. This is not a problem of any particular gender, race, or ethnicity. It's a human problem. And we're very good, by the way, at accounting and moderating our evolutionary disadvantages. We're born without fur. So we learned how to make clothes, we invented housing, and we tamed fire. On the website that's shown on this slide, you can find out what your own implicit associations are in 14 different areas. You may be surprised. I was. So I'd like to give you an example of implicit bias from an autobiography. In this segment of the biography, the author sees he's going from one place to another, and he gets on a plane, and he sees that the pilot is a black man, and he starts to panic. He says, how can a black man fly an airplane? So who is this author? Nelson Mandela. So as you can see, a black man is not exempt from having biases about other black men as well as other things. It all depends on how we're raised. Mandela was wise enough that he understood this and acted accordingly. I'd like to end by talking about stereotype threat. Stereotype threat is implicit bias turned against yourself. This is the topic of the book called Whistling Vivaldi. I highly recommend this book. It's short and it's easy to read. Stereotype threat occurs when a person worries that by their performance, they might confirm that they represent a negative idea about people in their group. About 40 years ago, Claude Steele, who wrote Whistling Vivaldi, began to investigate why Black students did not do as well academically as white students on average in this country when there was no reason to assume a difference in ability. This study, over the years that he worked, expanded to other situations where one group underperformed relative to another. For example, in this country, women tend not to do as well on math tests as do men. This isn't the case in other countries, Iceland, for example. And Dr. Steele did an experiment which has become classic. He took math majors at Princeton, a group of students that are very good at math, and he divided them into two groups of men and women and gave them a math test. The first group was simply told at the start of the session that they would be given a math test, see you in an hour. And then they were graded, and the men, in fact, on average, did better than the women. The second group was told that they would be given a math test, but although they might have heard that men did better than women on such tests, this test had been designed to eliminate that effect. And then they were given the test. But remember, it was the exact same test that the first group got. But in this case, the women did as well as the men. So what was going on? Well, it turns out that if a student cares about and wants to do well, but they are a member of a group which the society around them is providing messages that say that that group doesn't do well in that particular kind of thing, they might worry about their performance. We only have so much energy to use at any given day and any given time. So if someone is using energy to worry about the test that they're taking, they are not able to bring all of their energy to focus on the task at hand. They have divided their attention, and so they are not likely to do as well. And if you can ameliorate that worry, then it goes a long way to eliminating such performance gaps. And this is true in athletics, it's true in academics, it's true universally in anything where the society at large has decided, for whatever reason, that some groups are not as good as other groups in doing something. And for the students who are attending in particular, in your groups, through your peer support, through your teachers, you have to find and latch onto and truly believe that these biases, which you will suffer from the same as people who are not in your group, are affecting you, and you have to tell yourself, this is not relevant. This does not apply to me. It's a little bit the variation on the, it's not your problem, it's their problem. Well, they have made it your problem by making this culture where we think these things. And until those things are dealt with on a societal level, that's really what structural racism is the penetrance of all of these incorrect associations that we're making because we don't fully deal with our history. So with that, I'm going to stop. Thank you very much for your time and attention, and I'd love to answer any questions you might have. Thank you. I'm going to stop sharing. Bye.
That was great. I learned so much. My brain kind of is in overload myself. I'm going to turn it over to Morgan, who we have some questions here in the chat. She'll read them out for you.
Thank you.
Yes, thank you so much, Doctor. This was so inspiring and encouraging, especially with me wanting to go into or being a STEM student and going into that field. It's so inspiring to see women doing so many great things in this field. So thank you so much for coming in and talking with us and your willingness to answer our questions.
You bet.
So the first
really
question,
pleased to be here. Let's hear it.
yes. So the first question that we have is, if you could highlight one, which female strength would you lean into while working in a male-dominated specific group?
Well, that's a very good question, and I think the answer may be different now than it was when I started out. Remember, I'm old, and the world has changed a bit. So when I first started being the only woman in the room, which was pretty early on, the way I compensated was the following. I stopped wearing dresses. I wore pants all the time. Now I wear pants all the time. I had pants with pockets, so I didn't have to carry a purse, because some of the I got over that. I now carry a purse all the time, but then I would put everything in my pocket, because that sort of made them and me feel more comfortable in a group. And the other thing was I learned how to act. This is really important. You know, you've heard fake it till you make it. There's something to that. And that is, if you walk into a room with your shoulders back and you're walking in like you belong in that room, other people will tend to act as if though you belong in the room. And the magic thing is that if you do that often enough, you'll come to believe it too. So those are the things. And I think that maybe particularly as a Chicana woman, my family wasn't terribly communicative verbally. So I got real good at reading body language and what the things that were not said that were important to understand. That has been remarkably valuable to me. So, and I think women are a little bit better at that than men. Men just don't notice things as much, as my experience. Certainly my husband doesn't. So I think that's the thing, those are the things I would say.
Yes, thank you. Erica is asking, or is wanting to know more about how being a Chicana Native woman in the medical research industry provided insight that your colleagues of different identities couldn't have arrived at because of your identity and your view of the world.
In all the projects, I think that has come in handy. I mean, that was one reason, of course, that I was really interested in the diabetes project. My grandmother, my father's mother, died before I was born because she had diabetes that was not well treated, and she also had tuberculosis that wasn't treated. So I got interested in that early. We have lots of autoimmune diseases in my family. Everything from rheumatoid arthritis to multiple sclerosis and just tons. And so that made it interesting. And I think that the thing that I often bring to a conversation that's technical is the story behind the technicality, the story of people and the impact that whatever it is we're doing can have. So that's been important to try to pass on to my students and postdocs and to my colleagues as we look at different things. I had to learn, though, to speak up. You may find it hard to believe now, but I was not. I was a pretty shy person. I did not like to speak up, having come from a family that didn't, you know. But in graduate school, we weren't allowed, the students, first year students couldn't sit in the back of the room. We had to sit in the front of the room and we had to come up with a question. And it turns out that that's part of the acting thing, because, you know, we were told if you don't ask a question, you're going to get graded on attendance, and you also have to ask questions. So you had to think of a question. And early on, I learned that as reluctant as I was to ask a question, usually if I had a question, several other people had that same question. And so, and the other thing to learn is that think of the very worst thing that can happen. You know, if you ask for a particular thing, the worst thing that can happen usually is somebody says no. So okay, no. So what's the consequence of that? You move on to the next thing. So it really is helpful to think about the consequences of doing or not doing whatever.
Yeah, I think that's a really good point with coming up with a question, whether like when you're shy, especially, because I know for me, I don't talk a lot in class because I'm like, well, I don't really know what I don't know, so I won't ask a question. But that's a really good point and a helpful tip to, even though you may not know everything, find a question to ask, because someone else may have that question too, and they may be too shy to say anything. A lot of people may have that question.
A lot of people. And the other thing that's important to know is that everybody has insecurities. I remember coming back from Washington one day. I had been at a meeting there, and I happened to sit next to Phil Sharp, who is a professor at MIT and won a Nobel laureate. And this was after he had won the Nobel Prize. And he was really tired because he'd been at a meeting in Washington. So we were both kind of, ugh. And he said, so he loosened up and he said, I do not know how I would do it if it weren't for my colleagues. And he started telling me about the grants he hadn't gotten, the trouble he was having with people in his lab, the papers that weren't getting written or that he was uncertain about. Everybody has these insecurities. So that's another thing that we as humans share, is the ability to really worry well. So keep that in your mind too as you talk to and think about these individuals that are very impressive, but we're all just people.
Yeah. We have another question here. It says, do you think that women are more prone to exhausting their energies worrying about outcomes, or does that apply to any person regardless of gender being scrutinized or judged?
I think there's probably a tendency for women to feel more that way. We take more on ourselves. It's also true, I think, of gay folk. They have that side of them that is thinks in that way and worries more about the people around them. In some cases, not always. And of course, we can all name women who have absolutely not an iota of that kind of empathy. So it's averages. So I'd say on average, more women tend to wear themselves out. And that's a really important thing not to do. Because if you wear yourself out, we've lost you. And you can't do those things that we need you to do. And you are most able to do those things when you are at the table, as Hamilton says. You got to be in the room where the thing happens. There are steps to get there. And so you have to moderate yourself. You do have to take care of yourself. It's not, you're not letting anybody down when you say, I can't do that right now. I have these other things that I must do. And one of those other things may be taking care of yourself. And so what you say then is, I'm sorry, I would love to do that, but I can't do it now. You don't have to explain.
Yeah, that reminds me a lot of what my sister tells me. She's always telling me that self-care is not selfish, and you always want to make time to re-energize yourself, to recuperate, and you don't have to explain to anybody because no one is going to explain to you that they can't do it. They're just going to be, I can't do it, and go on about it.
Right.
Yeah.
And it is true that women, minority folk generally, have a harder time saying no when they're asked to do something. And this is something we really, really have to work to not do. We are entitled not to have to do everything. And we do ourselves no favor when we try to do everything. I always tell young women, you can have everything, just not at the same time. You have to make choices. Your priorities will change at different times in your life. And so you will change what your priorities. Sometimes it'll be family, sometimes it'll be jobs, sometimes it'll be other things, but it'll change over time.
Yeah.
And it'll change back.
Yeah, for sure. Do you feel like you've ever experienced a stereotype threat in your years as a scientist?
Yes, in fact, it's still true. I don't think, it's one of those things, it's like implicit bias. You don't really get over it. You just find mechanisms to try to deal with it. So we're all entitled to waking up at 4 o'clock in the morning going, what am I doing? Why did I do that? Or how am I going to get through this? Or how am I going to pay that back? Or whatever, how am I going to get that paper written? I've been trying to write that paper for 2 years now and nothing's on paper. This is not good. We can all do that. But in the end, you have to get out of that because that's a way of getting in your way of actually getting it done. Too much worry just takes away the worry from, getting the sleep you need to actually find the time to do the thing you want to do.
Yeah. What advice would you give to first-generation college students who are minorities?
Well, that's really, I think, that is truly the hardest job because you're negotiating a world that your parents are not well able to help you with because they haven't been through it. And so that means you have to get that help from other people around you, from your peers and from the faculty and from the advisors. And the worst thing you can do, you know, people will say, I'm too proud to ask for help. I don't need anybody's help. I'm supposed to do it on my own. Nobody does it on their own. Everybody has had a lot of help. Some of them don't realize the help that they've had when they have, you know, I feel I was privileged in my college education. I had a head start because my parents had gone to college. They were the ones who had to make that first hard step and figure it out. And so that's something that as a first generation student, you've taken on a big, important, critical thing, both for yourself, and it's a remarkable how what the effects can be. My dad, as I said, was the middle child in his large family. All of his older siblings didn't go to, they stopped schooling at about the third or fourth grade and went to work. So, and their kids, it's mixed, whether they went to college, some did, some didn't. But after Dad went to college, all of his younger brothers and sisters went to college, and all of their children have gone to college. And the children, my cousins from the older families who did go to college, always talk about Dad as being the influence on why they did that. So you're changing generations just by being what you are today. So you shouldn't forget that. And you really, there's nothing wrong with asking for help, be it financial, psychological, any kind of thing, because it's a little, you know, it's a battle. And so in order to win that battle, to get through it and get to the next stage, you got to finish. There's a big difference between starting and finishing. That's why I think my sister's story, Dorothea, because when they eloped and went off to California, we thought, you know, we were all wondering what was going to happen. It took them 10 years to get their first degree because they were doing it part-time and working full-time. But it all ended up very well. And now, you know, my brother-in-law has his, well, they share a Ferrari. He has his Porsche, and she drives her Honda because the company gives her the Honda. And they were the first in the family to have one of those great big TV screens, flat screens. I hadn't seen one until I saw my sister's. So it's a tough thing you're trying to do. I know it is, but the payoff can be enormous. It's really important.
Yeah, and sticking with the changing of mindsets and generations, Sunny is asking or is saying that many Chicana women in her life feel that they don't deserve more or deserve greater jobs. How can we change that way of thinking?
Well, you know, one of the truths of life is that sometimes you can't change someone's mind that you really care about. And so there's two ways to go about dealing with that. One is you keep trying, you appreciate them for what they are and what they have done and can do, because all of them are doing their best. That's one thing that we've learned about each other. And in my family, for example, is that we certainly have a lot of different opinions and a lot of different life tracks. But basically, we respect and admire and love each other. And so when someone doesn't feel they deserve something, and I know it's true in the families, you have to give them the same pep talk that you're giving themselves. And you also have to not make them feel bad about themselves. One of the best ways to help someone think about something new is what you do. So that I remember there was a program at El Paso, at the University of Texas, El Paso, where someone decided that really, if you want to get kids thinking about college, you have to start in grade school, which, by the way, I believe. And so they started bringing these kids from the barrio into UTEP just to have lunch in the cafeteria, to meet professionals who look like them from around the area. And the mothers and the daughters, at the end of their time together, the mothers would come too. And now there's a program called Fathers and Sons. It does the same thing. They had to sign a contract to each other where the daughter would promise to do their homework every day and stay in school and not get pregnant. And the moms had to promise that they would provide a place where the kid could study and would support them through their studies. And what was remarkable was that not only did all those kids, I think, you know, in El Paso, back at the time I was aware of working with the program, high school students in El Paso had a huge teen pregnancy rate, really big. It was a problem. And of the 300 girls that were in these first several cohorts, 2 of them got pregnant before they graduated high school. Whereas in the general population, a cohort of 300, their peers who didn't have the program, something like 70% of those kids got pregnant before they got out of high school. So this had a big effect on them. And the moms went back to school, about a third of them. And the other moms that didn't go back, still, they started thinking about, you know, what they could do in terms of jobs or learning English or whatever. So your example is something that can influence the older folks in your life who might be underestimating their own capabilities and futures. It's hard right now. This pandemic has been very hard on everybody.
Yeah, it's been extremely difficult for, especially I know for me, my science related classes. It's so hard doing it like online versus
Oh,
hands-on. It's a
very
nightmare,
hard.
but
And most professors haven't learned very well how to deal with an online kind of class. It takes a different sort of approach to do it. And so, and you know, and teachers are busy too. So taking the time to figure out what the best practices are is daunting.
yeah. And we have a couple more questions here for you. You mentioned a professor said that women do not belong in chemistry, and that a college you applied to was not accepting women. And we all know that that mindset and that way of society wasn't that long ago.
Yep.
Do you think those same ideologies still exist at present, in the present, despite efforts to eliminate that type of thinking in STEM?
They do. And I've concluded, especially in the last 4 years, when we've seen a rise in these kinds of attitudes, I think that the message that comes that the president gives just filters throughout the population, and it gives permission for people to do and say things that previously, and I hope again now, are simply not socially acceptable. So people are more willing to act out. And that's not good, because Martin Luther King once said that I don't care what somebody believes or thinks. I care how they behave. I want them not to do any more lynching. That's why there's been such a push to try to get a law, because there isn't yet, you know. It's up there, but it hasn't been passed. And so when we have a society that says these kinds of things are not said, we're going through that right now with the Me Too movement. We're being very hard on men, whether in a very large spectrum of sexually inappropriate activities, you know, from a rubbing the back to rape. We're treating them pretty much the same way in terms of how we're reacting. And so we'll swing. We need to figure out the happy medium, and we need to make the message clear that from the inappropriate touching to the other end, it's not societally acceptable. And my belief is the following: if we could actually make people behave properly, over time, people who truly believe that stuff, but couldn't act on it, grow old and die, right? And the younger folk who have a different experience then grow up, and you see less of that. And I can say that as now, this is a very long battle, right? It's been going on for 100 years in this country. And I've been part of it for maybe 45 now. And so I can say, yes, there have been changes. There are things that are possible now that weren't 40, 50 years ago, which is not to say we're anywhere near where we need to be or want to be, but things can change. It takes constant pushing. I think that the recent activities with the Black Lives Matter, with the Asian attention to Asians being attacked, with Native Americans all of a sudden, you know, being very highly positioned in the administration and being able to make big changes is a thing which is going to help change things. But it's slow, and we backslide because these implicit attitudes are so hard to get rid of. We can only modify the way people behave, and we don't know yet what the best practices are to try to change that kind of behavior. So it's getting better, but it's not yet where we need it to be. And it feels particularly bad now because we're all so separated. You know, it's just been a terrible, awful, no good year, any way you look at it.
Yeah, we've come a long way, but still got a ways to go.
Right. I see a question here from Kristen that says, what advice would you give if I feel more comfortable in a typical feminine attire? I say go for it. As I said, I'm old, and so the attitudes were a little different there. And I think now there's much more freedom, both for men and women, to pretty much be whatever they want in their dress. You know, you sort of, I think the best hint is to say, when you go into a new situation, you know, when I visit a university or something, I dress up. It's true I wear pantsuits, but I do dress up. It's a symbol of respect to my hosts. And so when I'm going into a new situation, I'll tend to overdress rather than not, just because that's safer, frankly. On the other hand, so in Chicago, that's a city where people dress up. So in Chicago, I did even wear a skirt and a blazer or whatever now and then. And there were times when I had to really dress up because in my role, I did everything from go to football games where I had to schmooze the trustees to formal events where, you know, you wore formal clothes. That was fun. I actually like clothes. So I think if you're more comfortable, one of the most important things, despite this business about certain things you can act, it's really important that you come to know who you are, what you like, how you feel most like yourself. And then that's the way you dress. And remembering that people take note. And so your dress should be appropriate to the situation, whether or not it's pants or a skirt. And certainly makeup is something that both men may not notice it, but they notice when it's not there sometimes. So, you know, I have friends who could not tell the difference between having mascara on or not, believe it or not. But if you go around with no mascara, they say something's different. So that's and women, of course, notice instantly. So you could certainly wear your favorite red lipstick to an interview, and a pencil skirt and blazer is perfectly appropriate for any board meeting these days. If you're in a biotech company, if the CEO wears blue jeans, that's okay then for you to wear blue jeans. But when I go to ST board meetings, I almost always wear blue jeans, but I usually wear a blazer with it because the firm out in Navasota it's a ranch. There's bulls and barns and mud, so we all dress for that. So that would be my answer to Kristen. So the question was, I had to change how I dressed and acted. So would I say that I had to toughen up? Oh yeah, big time. You do have to toughen up. That's part of growing, I think, generally. I think that that's one of the reasons that the nice thing about young people is that they are still very passionately involved and should be, because we need people who are, you know, good time troublemakers as well as people who are negotiating through the bureaucracies and so we need all of the entryways we can get. And so you have to do it according to who you are and what you think you want to be. So to change how I dressed and acted, it was hard. Sometimes that's all it was acting. You know, I was saying, ugh. But as I said, I think the most recent example is there's a committee I'm on at the National Science Foundation. It's a committee that's working on ways to increase representation throughout the sciences at a national level. And as part of being on that committee, we have to be, each of us chooses another part of NIH where we sit on their advisory committee. And I chose the Committee on Social Sciences, Economics, and Behavioral Science because I wanted to learn more about it. So when I got there, I'm the only I'm not a sociologist, I'm not an economist, and I'm not a behavioral scientist, although I was a neuroscientist at one time. Still, I am really a fish out of water. I'm not the only minority person on that board, or even the only Hispanic, because that committee is primarily that kind of people, because that's its job. And it's still true that we're the ones who are most thinking about that issue. But still, the inclination to say, I do not know what I'm talking about in this group. These are the people who are experts in this thing. What do I know? But very quickly, because it's who I've become over time, I started being a participant. And so now those folks consider me a part of their group. I even got appointed to a second term independent of my other appointment because it's an example of how someone who's different from everybody else in the room brings a new perspective. I'm bringing the perspective of a natural scientist to a bunch of social scientists, and that interaction has been really fruitful. And so part of what's happened is that the group has decided it needs to meet with other committees from the different places to try to get NSF parts and pieces to work better together. So that was a really good thing to do.
Yeah, that's amazing. I do have one more question for you. Jason is asking that what can men do to support more women in STEM?
This is a good thing to do, is that to be an ally. And you can do it as a man supporting women in STEM, and also people of different backgrounds supporting other people of different backgrounds that are not the same as their background. And the way it works is as follows. You'll notice that most of my mentors and supporters were white men. All but Loretta, really, of the folks who have been most important in advancing me through my career have been white men, aside from my family. And that's important. The reason that I want to point that out is because people will be different. So, and the thing that made those men stand out, some of them are real tough. You know, they're not exactly cuddly guys. They were very demanding. But the core thing about them was that they were demanding of everybody. They truly did not make a decision about what I could do or could not do based on who I look like. And I picked up on that. At the time, I could not have said that. What I was feeling was, what I felt was, I cannot have a decent conversation with so-and-so. I just don't want to talk to him. It was usually him. Whereas this person, who was as smart or smarter than that other person, and who was very critical of what I said, I could talk to them. I could learn from them. And that's because at their core, they had not decided what I could do. They were waiting to find out. And in the end, they came to appreciate what I could do, because when we got to know each other, when they saw me, whatever I did. So to be the best ally, the first thing that you have to know about is that you believe or are willing to work towards the belief that women are as capable in STEM, and that when you see a woman struggling, you will say something to the men who are being inappropriate. And that goes such a long way. And you get to know a little bit about how it goes. You know, what is the life of a woman in STEM? There's a documentary out now called, I'm going to forget its name, but I'll remember, and find it in my notes and send it to Carmen. It's about 3 women scientists and how the things that they went through. And it varies. It varies in age from a little bit older than me to a beginner. And some of the things they went through, from sexual harassment to unfair allotment of resources, not as much lab space as other men, underpaid, all that kind of stuff, and how they went about dealing with it and what their allies did. It's very much the same as you have to know a little bit about a woman's world, which means there are books you can read for sure, but mostly you can have friends and talk to them about this in a place where you both feel comfortable being quite honest with each other about what life is really like from your point of view and from their point of view, and learning about each other. This is something that I think we don't do enough of. So to be an ally, you can, in your lab classes, pick a woman, be a partner with them, support the women. When your presentations are going, truly take turns doing those presentations. Don't assume that you, the man, needs to do it all the time, or that you should defer to the woman all the time. Real partners are just that. They're comfortable or willing to try, at least, to be both supportive. And sometimes supportive means pointing out errors in behavior or just plain mistakes in the lab processes you're going through. So that's one way to do that. And I'm trying to find something here. Oops, she might be gone. If I would like to say, and Carmen, you can tell this to people, I am on LinkedIn. And if you contact me, I will respond. It might not happen right away, but I will respond. And just remind me that this is where we met, because, I can't see most of you. So I don't know what you look like even. And I have a terrible memory for names. They just fall right out of my brain. When I was dating my husband, I would, go I'd go pick him up because he was working at the health center, and I'd flip open a textbook and say, oh yeah, Tony. Okay. I'm bad that way. It's just those neurons never got developed in my head. So.
Well, I want to thank you for this well-informed, impressive, and empowering presentation. I almost forgot where I was for a second, and I was like, can we give it up for Dr. Lydia Komaroff one more time? But we can use our emojis, y'all. So if y'all see the comments in the corner, please, that's going to be our round of applause.
Oh, thank you. Thank you.
I wanted to go over just one more thing with y'all participants. I'm going to share my screen here just really quick. Again, this was our first Women Chat Wednesday speaker series for the month. But our next one, let's get out of here. Ooh, come back. Uh-oh, come back. What happened? Here we go. Yes, our next one will be on March 24th. And our speaker will be Andrea Wise, and she will be talking on the power of self-care. So please join us on March 24th at 1 p.m. And also, of course, to register for that event, please feel free to go to dccd.edu/women's history. That's where you'll find all of our events, the time that they start. And of course, you can register for our Women's Chat Wednesday speaker series. Also, if you enjoy this, or even however you feel about it, please make sure you are filling out our survey. We're going to drop the survey link in the chat. Oh, we already done that. Great. Thank you, Erin. Please fill out the survey for us. And of course, if you can find Dr. Lydia Komaroff on LinkedIn. If you would like any other information, you can always email myself. And I hope you all have a phenomenal spring break, because I know that starts this weekend for a lot of us. So, and I just wanted to thank you so much for coming and celebrating Women's History Month with us. And a great job again from Dr. Lydia Komaroff.
Thank you. Thank you again. I truly enjoyed it, and maybe I can come visit you sometime when all of
Right,
this is
face to
behind
face.
us.
Thank you all so much. If you want, Dr. Via, Komaroff, again, I know you said you're free to stay till 3:30. Students, if you have any other questions, we're here for 9 more minutes.